So the field of post-parasite coevolution is well explored in biology, and it has immense importance in various aspects. For example, in medical field. But what is little less explored is the environmental effect on post-parasite interaction. So I'm going to talk about this, but before that, what I would like to mention that parasite has like immense implication on the fitness effect on their host, and therefore may be represented as a potential agent of natural selection. Now the question is, if that is the case, what we can expect that over time all alleles related to host resistance can get fixed. But what we see in general in nature that maintenance of heritable genetic variation for resistance. Why is it so? One potential reason could be that the existence of cost of resistance. That means that fitness reduction associated with resistance in the absence of parasite. The cost effect in this case, life history cost effect, is coupled with variable genetic variation, which can ultimately lead to the maintenance of genetic variation for resistance. There are studies showed that this life history cost effect exists both in ecto and endoparasite and also in plant and animal. So today I'm going to focus on some major hypotheses. One is if there is any cost related to ectoparasite resistance in terms of their lifetime fecundity, lifespan, and larva to adult survival, and whether that cost effect modulated by different environmental conditions. So this is our model system, Drosophila melanogaster mime and Gamasodes, Drosophila melanogaster host and Gamasodes mime. They are naturally coexisting in nature. So here you can see that the kind of interaction where a mite is approaching a fly, and then that mite is sort of exploding whether to infect or not. And at this stage, host is really showing that immense avoidance behavior, for example, kicking, and that host can get rid of mite, but sometimes they get infected as well. So this model system was collected from Northeast Australia, and this is like one of the collection places, as you can see, and this is the jackfruit. This is here they exist. This is their natural habitat, that mite and flies. So we collected them from here, and now to express the cost, how to assess the cost. Of course, there are several avenues to assess the life history cost, and one of them is to run artificial selection and followed by contrasting your control and susceptible genotypes in terms of different fitness traits. How you do that? You start with a large population of ample genetic variation and then expose those hosts or flies to the parasite and just select uninfected host and breed them to the next generation. You keep doing that until your selection responds, followed by testing your cost hypothesis where you can contrast control and selected lines. So this is like the more details as I explained before. So it's all about artificial selection. So I would like to move on to the result slide. Here, 3 blocks, line 1, 2, and 3. So we have 3 different lines. So blue lines are the selected lines, the red lines are the control lines, y-axis is the proportion of uninfected flies, and the x-axis is the generation. As you can see that we had like treatment main effect. That means over time these selected lines, they showed they had higher resistance in terms of avoiding mites. And also this treatment generation by treatment effect, that is also significant. That means that they diverged over time, that at selected lines were more able to avoid mite more efficiently. So to details of this experiment, today evening at 5:30, please visit Mr. Harman Singh's poster number 197. So now we focus on different life history traits. So one, female fecundity, adult female lifespan, and larva to adult survival. So here is the experimental design. So we had 3 lines, resistance line and control line. We tested them, as I mentioned before, that environmental modulation we wanted to see. So we tested them under 3 different environmental conditions. One is low temperature, medium temperature, and high temperature. And what we did, we took 10 females from each temperature regime and from each line. And from each replicate female, we put them in a food vial, allowed to lay eggs, and over time we transferred them, and at the end we counted for total lifetime fecundity and lifespan. So here is the result. So here that and we did that at generation 16 and generation 21. We counted for lifetime fecundity. Here the y-axis shows the total lifetime fecundity. Dark gray is the control lines, and the light one is the selected lines. As you can see, and these blocks are the different temperature regimes. As you can see, the temperature main effect, the selection treatment main effect. That means that consistently across all temperatures, that selected lines had lower fecundity. Correct? So, however, we did not the same design. We did not find any cost in terms of their lifespan. So, so this question was tested previously using a completely different fly a mite model system, a desert fly and macrochelis mite. Here you can see. So this is the fly infected with mites. They are collected from Arizona, and this is the result. Here y-axis is the lifetime fecundity, and they were tested under two different environmental conditions, low and high temperature, and selected lines had lower fecundity, lifetime fecundity compared to their control. That means the resistance cost in terms of fecundity is pretty much consistent across two completely different fly mite model systems. So now move on to the pre-adult cost. So in this case, we tested their larva to adult survival under ammonia concentrations. Why ammonia? Ammonia is their natural excretory product, and over time that ammonia build up and create more stressful environment to them. So here we tested our 3 lines, and we had 4 different ammonia concentrations: 0, low, medium, and high. So that is the pre-adult part, and whatever the females flies survived, we took the females and put them on the fecundity assay. So, and again we tested 2 different temperatures, and at the end we counted for total fecundity and lifespan. So this is the first part. This is the pre-adult survival. Y-axis is the larva to adult survival. X-axis different ammonia concentration we tested. As you can see here, we got like selection main effect significant. However, we did not find any concentration by treatment interaction effect, which says that okay, the selected lines have lower survival but consistent across all. But our post-hoc analysis actually revealed that in the 0 concentration there was no difference between control and selected lines, whereas this effect was consistent across low, medium, and high ammonia concentrations. So this is the second part. Female fecundity. Here y-axis shows the lifetime fecundity. So here X is the female came from either 0 concentration of ammonia or medium concentration of ammonia, and here 2 different temperature regimes, high temperature and medium temperature. So for now we'll focus on only high temperature, and again here we found like the treatment main effect, but and there is no interaction effect between concentration and selection treatment. But again here our post-hoc analysis revealed that the difference is way more magnified when female came from medium ammonia concentration and exposed to high temperature. So we wanted to see the molecular underpinning of this fecundity cost associated with mite resistance. So we did RNA sequencing. So and in that we counted for differential expression followed by mapping gene ontology. So for today's talk, I'll just focus on the flies were not exposed to mite because that was relevant to our cost hypothesis. We did 2 different analyses. One is like the combined analysis, all controlled versus all selected, and the paired, that means C1 versus S1. So these are the paired, the number 2, where lines were compared. So we see that differential expression of many genes across lines, but there was many less overlap if you really compare line 1 versus line 2, line 2 versus line 3. So, but from our general comparison, we see that there are genes differentially expressed. They are mainly involved with stress response, ATP metabolism. They are the defense mechanism, etc. So that means that here we identified some potential genes involved in the fecundity cost, but we haven't validated that. We need to do functional validation of these genes, which can be done later on. So with that, I summarize that the main thing, the first thing what we found, that the fecundity cost associated with resistance. The second thing is that fecundity cost is specifically magnified by pre-adult condition of pre-adult stressful condition, and also temperature effect, adult temperature effect, and that cost, fecundity cost, is pretty much consistent across two different fly and my model systems. And finally, we identified some candidate gene that can be related with their fecundity cost. With that, I would like to thank my supervisor, Professor Pollock, and Dr. Josh Benoit, and the entire member of Pollock Lab. Harman is going to present the poster today. So he was really involved in this project. And Parker, so thank you all. Yeah.
What is the, do you know the actual parasite load in nature?
Yeah, it can go, I mean, I don't know actually nature. So, but in laboratory system, when we infect them, it can go up to 9 or 10. I mean, then those flies they cannot even move.
Right.
So they cannot even move. I mean, they just simply give up.
But I just wonder under natural conditions with the fruit that they're using, and whether they can avoid them in a different way that they could in a laboratory, things like that.
I mean, in laboratory, I mean, what we see, I mean, as I mentioned, that they are really like they're kicking them, and also Josh Benoit actually did some sleep cycle assay with that, with this selection line. So what this data I did not present here, but what he found, like they are having like very less sleep. So that means that as soon as they sleep, they are getting hit by mites. So they are having very less sleep. Yeah. So yeah. Any further questions? Yeah.
What was the reason behind doing different temperatures?
Yeah, because the temperature can modulate. Sometimes the effect of cost can be present, but that cannot expressed unless you put them in a stressful condition. In our case, the temperature was a kind of stressful condition to them because we put them in high, medium, and low temperature, right?
Does it affect the parasitism rates, or?
Well, parasitism rate in the sense, I mean, here we are talking about the temperature effect on the fecundity cost, not the parasite.
Yeah.
Yeah, so the temperature effect, whatever I showed, that was when I was talking about fecundity cost.
Yeah. Okay.
Yeah, not the parasite. Yeah.
Right of that. What was your base population for selection?
So the base population was the that was the population collected in Australia, the city base.
Okay, so some of those animals have actually seen the parasite.
Yeah, of course they. Yeah. That's what I mentioned. That that was that Grace Food Farm. That was like one of the collection places. So and they naturally coexist in nature. So they were collected by Professor Pollock in Northeast Australia. Yeah.